Yes, now you can ask your doubt.
So in section 15 and 16.2, we are source of the information is signal management team, right?
Tracker. Yeah.
Tracker. So from there we copy pasting or we are doing some changes in aggregate report.
So see, in section 15.1, we basically present the tabular format of whatever signal is ongoing or closed during the your reporting interval. Okay. Ongoing means it might be the evaluation might have been started during your reporting period, or the evaluation is ongoing from the previous reporting period, but the status is still ongoing, means the evaluation is not complete. Okay. So this tabular format you will present in section 15.1 from the signal management tracker, exact copy paste, it will be there. And then you will say that the complete log is presented as an appendix. There is a signal log that we provide as an appendix. So either you can state in 15.1 that all the new ongoing and closed signals during the reporting period are presented in appendix 3, or you can say write the tabular format in 15.1 itself. It's both ways, it's fine. Okay, because any which ways you have to attend the attach the appendix. Then in 16.2, what happens? Whatever signals are closed. So if the signal is closed, then there will be a signal evaluation report prepared for that. Okay. So you have to take those signal evaluation report and you have to copy paste from them only. No need to evaluate anything. Okay. So you have to copy paste from there only, but a limited information like why this signal was initiated, what sources were uh, what sources were evaluated in this, what was the discussion point, means what was the conclusion of that, and finally, means discussion means whatever you find, whatever was the finding from that. So that will be there in the signal evaluation report. And finally, what was the conclusion? So these topics you cover in that closed signal part. Suppose there are 4 signals closed. So in each signal, you will take their signal evaluation report and present this information in 16.2. Apart from that, you will mention that if there are 4 signals, so you will mention that all the 4 signals are attached as all the 4 signal evaluation reports are attached as an appendix to this. So you will attach as an appendix that signal evaluation. So since we are attaching appendix for both, so we are not presenting or we are not evaluating anything. It is directly from the signal. Now, in section 15.2, which is not basically a signal, it is either a regulatory authority request or safety issues under monitoring. In that section, you evaluate the reporting period information as you do for important potential risk. Is it clear now?
So the section for section 15.2 PSUR tracker, from there we will take the information.
Yes. Your source will be PSUR issues tracker. Whatever regulatory authority requests are there, whatever safety issues under monitoring, they all will be present in PSUR issues tracker. So you have to be clear, there is a signal tracker, there is a PSUR issues tracker. And from where you have to take the source for which section.
So this 2 section only in 15 section 15.
I'm sorry, this 2 section means?
So in subsection I'm talking about, like in section 15 there are 2 subsections, right? First is signal, then
15.1.
second one is request from health authority.
Right. So for 15.1, you will take from signal management tracker. For 15.2, you will take from PSUR issues tracker.
So from 15.1, I'm asking about the format, like whatever format is there in the signal tracker, we'll copy paste it from there.
Yeah, the same format will be there because the signal tracker is actually made on that basis only. Means in signal tracker, what would be there and what information you need to present in 15.1. You need to present what signal, the name of the signal, on which date it was detected or validated, whatever that is there. It will be mentioned date opened as. Okay. Then what was the trigger for that signal? What is the status currently? And if closed, you can provide the conclusion, but no need. If you don't provide the conclusion here, then you have, if it is closed, you have to provide everything in 16.2. So no problem. And then if it is closed, what is the date of closure? So this is the tabular format that we use for 15.1 mainly from the signal tracker. And this all will be there in the signal tracker.
Hello, Amit sir. See, means I have one doubt. In section 15, right, we have to write only ongoing sections, or we have to write new, ongoing, and close. All three we have to write. And again mention close in 16.2.
The 15.1 will present new, ongoing, and closed. Any which ways, if there is some new signal, then definitely it would be under some stage, either validation or so it is a ongoing stage only. In signal
Okay.
tracker, you won't find it as written new. The status will be ongoing.
So all three we have ongoing and close. We have to both mention in 15.
Everything is 15.1, and in 16.2 only closed. The summary of the closed signals from their evaluation report.
Okay. And my second point is, sir, when we are writing 15.2 section, that is RA request, right? So what are that RA request belong to? Are they risk? Are they signal? And how do you evaluate them? What we have to write about them?
So if there is any risk or signal, then they have different sections as we mentioned. Okay. So RA requests are like suppose EU authority asked you in this reporting interval or maybe earlier also, no problem with that. So they asked you to evaluate the cases. Example, I'll give you an example from scratch. Suppose EU regulatory or PRAC, you can say. PRAC is, always remember what is PRAC, okay? Um, so PRAC has requested the MAH to present the analysis of a drug event pair. Suppose azithromycin with thrombocytopenia. Now, as an MAH, listen to the story, okay? Complete story. Now, as an MAH, I evaluated azithromycin with thrombocytopenia, but I was not able to find any causal association between those, and not even the sufficient evidence from either the literature or from my spontaneous or my database. So in that case, I am in confusion. I'm not able to establish a causal association. I'm not able to find any sufficient evidence so that I can at least say if no causal association, then I should make it a potential risk, important potential risk. Okay. In this scenario, I'll reply to the RA, health authority, with whatever information I found with my signal evaluation report. Now, health authority comes back and says that, okay, no problem if you're not able to find, but keep monitoring the issue, monitor it. So in this case, this is not my signal now. This is not my risk now, right? So it is not in my RSI also. And health authority has asked me to evaluate or keep monitoring. So in this case, what I will do? So for these cases, we have section 15.2, where we can present our monitoring issue. Like what will happen? I'll keep a heading of thrombocytopenia in 15.2 under regulatory authority request or health authority request. Now, in my current reporting period, whatever cases I have received for thrombocytopenia or from my search criteria, those cases I will analyze and present in this 15.2 under thrombocytopenia. How to analyze those cases? The same process you have to follow as you do for important potential risk in section 16.3.2. Now, am I clear?
Yes.
This is one part. Second part is if you have made some certain risk or certain, you can say certain topics as a safety issues under monitoring. So those also will come under this section only and will be evaluated in the same manner as I discussed for the previous. Now, am I clear what it goes in section 15.2?
Yes. So means there are normal, like there are some normal ADRs which we have to evaluate, right? In
We
15.2.
don't have to use the layman terms here. No normal ADRs, nothing. If it is significant, then only we are monitoring it. Why is it significant? Because health authority asked you to monitor it. So it is something significant. They could have find from some other MH. So that's why we have to monitor it. Some issues you as an MH has kept under safety issues under monitoring. So it is a significant safety issue, not a normal area. That's why you are considering it for monitoring. It has a potential, means it is something significant because there are many areas listed in RSI, but we don't monitor everything, right?
Yes.
But this is something significant safety, which if turns into Like if you are able to get some evidence or if you are able to establish the causal association, then it might change into an important potential risk or important identified risk. That's why we keep these ADRs, you can say, or these potential risk means these ADRs, you can say, or these drug event combination under safety issues under monitoring, right?
Hello, Amit sir. Are there any factors means in whatever significant terms we mentioned in 15.2, are they becoming signal or risk further or in that scenario?
You see, again, you are confusing. It was already a signal. That's why you have made it either safety issues under monitoring or you have evaluated and health authority has asked you to monitor since you are not making a risk. Okay, so it was already a signal before coming into 15.2. Now, whether it has completed its journey from signal to risk, that is not yet completed. Okay, so once you have started monitoring it under 15.2, and if it is completing its journey for risk, then it will eventually move to the other sections. So currently, it is not a risk, but it is a significant safety issues to be monitored. And before coming to 15.2, it was a signal which was evaluated, and there was a signal evaluation report submitted for this. Okay.
Yes. Amit sir, I have one more doubt. I don't think it is really one doubt, but I just have one doubt. That's why I'm asking. See, whenever signal is converted into risk, right? At
Hmm.
that time, we add it in RSA, right?
Hmm.
So, in RSA, what is the situation where we only add it under adverse drug reaction? How the adverse drug reactions are added in the RSA? On what basis?
Okay. See, there are different sections in RSI, like we
Yes.
studied 4.3, 4.4, 4.5, 4.6, 4.7, 4.8, which are basically talking about the ADRs and risk and everything. Okay. Now I'll tell you one scenario. Suppose if a health authority asked you to evaluate the same, that what we discussed, azithromycin and thrombocytopenia. You evaluated it, you presented your signal evaluation report to the health authority saying that we were not able to means establish a causal association or find a sufficient evidence. That's why we are refuting this signal. Now, health authority comes back to you and says that suppose you are Pfizer as an MAH. So they says that we have other MAH like AstraZeneca and Novartis who has evaluated and included it into the RSI. This is the wording for RSI. So request you also to include it into your RSI. Now health authority has mandated you to include into your RSI. Okay. But you are not able to find sufficient evidence or you are not able to establish the causal association. So you will reply to the health authority saying that, yes, we are including it under section 4.4 with the wordings that you have provided. Okay. In this case, you will include this risk under section 4.4. However, you don't have evidence and nothing, but other MAH are having and health authority has mandated you. So that's why you will include this risk of thrombocytopenia under section 0.4 and your RSI will be updated in this section. But you will not update 4.8 because 4.8 is basically the areas that you are getting into your database or from literature or whatever sources you are having some evidence for those ADRs, right? That's why those are listed in 4.8. But for this, you don't have any evidence, you don't have sufficient evidence, or you don't have any other thing to suspect even the causality. But due to health authority mandation, you are including it under section 4.4. Clear?
Okay. I mean, sir, also when one signal is closed, right? So that also sometimes reopen also. Means we
Hmm.
are, if signal is closed, then also we are tracking it or what?
If signal is closed, we are not tracking it. But what happens, like suppose in this case, thrombocytopenia, you closed with no any further action. Neither you have made it safety issues under monitoring, neither health authority has mandated you, neither you have made it risk. So it is refuted and closed completely. Now what happens? After 1 year, you start getting more cases of this thrombocytopenia. So it will flag as a signal. So in that case, you will go back to signal management tracker, check whether it was evaluated earlier. So what was the conclusion? And again, you will evaluate post that reporting period. You will evaluate all the cases that you have got, and you will give your conclusion as to what you think from this signal, which is flagged. So it can be opened, no problem with that. But you will see the previous history as to what was done with this signal.
Okay. Sir, also I have one doubt in the section 16.4, right? In that we take cumulative data.
Hmm.
And any other section is there in which we take cumulative data?
Section 16.4 onwards, all the sections include cumulative data. 16.4, 16.5, 17.1, 0.2, 0.3, 18.1, 18.2.
And whatever we write in section 6 and all, means whatever about serious adverse event from clinical trial and all, we only take from interval.
Serious adverse events, these things are taken mainly from interval, but that serious adverse reactions that you are presenting in section 6, so they are giving you a summary tabulation. Okay, so you don't write there. They give you a summary tabulation. In that, both information is there from cumulative also and interval also. But that you don't evaluate, though you don't write. That is you can say ready-made section coming from the stats team.
Okay, so we directly get, we don't calculate.
Yeah. So, but in that, all the serious adverse reactions will be there from both, and they will be differentiated from interval reporting period and cumulative reporting period.
So the summary also they give, or we write the summary for it?
You don't need to write summary for those. If you will see, you will just refer to the appendix.
Okay. And sir, for whatever it is there in section 7, like completed clinical trials are there and ongoing clinical trials are there, right? So in that, the clinical trials are from starting, right? From IBD. So at that time, we write all the information from cumulative, or how we present it.
So, have you seen any PBR, any sample PBR
Yes,
for section 7?
that we exhibit only I have seen.
I told you to leave that as an example. But always remember, since you have asked, I will tell you the summary of you are providing the ongoing and completed clinical trials during your reporting period in section 7, right?
Yes,
What's the heading for that section 7?
completed clinical trials and on summary of significant findings from clinical trials during the reporting interval.
And there will be two subsections. One is ongoing,
Yes,
one is completed, right?
completed. Yes. And third
So
one is long-term follow-ups.
that is a different part. First, we consider the clinical trials for ongoing and completed. Okay. So what happens? You present a tabular
Sorry,
format.
sorry. I'm sorry to interrupt, but I don't think we have completed these sections, right? 7 and 6 and all.
We have not completed, but he is asking, so let me reply. We'll
Okay, so actually, I was wondering whether we have not completed it.
no,
That's
we
why I
have not
asked.
completed, but he is asking, so let me reply. So what happens? That first in ongoing and then second part is completed. So you have to present a tabular format. In tabular format, I discussed this in previous some class, I remember. So there is a tabular format in which you present what is the title of that protocol or the study. Okay. Then you will present what is the description of, like it is a phase 1, phase 2, phase 3, phase 4, which study it is, then whether it is randomized. So there will be a title of protocol that you can find directly from the clinical trial information that you are getting. Okay. Then you will provide what is the method used to analysis. And then what is the status of the study? Like if it is under ongoing, then definitely it is ongoing, but still we write status as ongoing. And last section will be any significant safety finding. So if there is any significant safety finding, from where you can get that, it will be from the CSRs for that. If it is ongoing, there will be interim CSRs presented in between, not the final one. So from the interim CSRs, if you are having any significant safety findings, you will write there. So in this way, you will present all the clinical trials ongoing. In the same way, in completed, the same tabular format is used. We use the final CSR for, because if a study is completed, then final CSR is up. And what is final CSR? That is stamped and signed. So we will take information from final CSR. If there is any significant safety finding, we will present there. And it is endorsed by or reviewed by the clinical science team. Okay.
Okay. Sir, one more doubt. In section 9, right, whatever it is 9.1, right, other clinical trials. So for the other clinical trials, from where we take the information?
Actually, Other clinical trials are the ones which are not sponsored by your company or which you are not aware of, which for which you are not maintaining database. But you get some information from your epi team or some your non-clinical team or your clinical team about those. Those will be presented in the that is a part of the CS team only to provide you. But if they are not providing and you are some other source, the client might be having some other source also to track all those things. So from there you will get this information, but ideally 9.1 is not applicable in all the scenarios. You can say that you don't know. Means you have not encountered any scenario in which you have presented 9.1.
Rami sir, what about section 10, non-clinical data? In that, is there any things which we get?
So Tanmay, now I think people are
Okay, fine.
getting confused. So you can ask me all these things later on because we have not covered this section. You can get me in touch with me separately.
Okay, fine.
I'll discuss with you, no problem. Any other questions, people, from the previous sessions that we had?
Hi, can you hear me?
Yeah.
Yeah, so section 15.2, that information for signal, it will be in the 15.1 as well, right?
No, that is not a signal. If it was signal and changed into safety issues under monitoring, then it will be under 15.1, but mostly it will not be there in 15. It is safety issues under monitoring or health authority topics that might be coming from the previous session, means for the past 2-3 PBRs you have been evaluating it. So it is not necessary that it will be there in 15.1. If it is evaluated or open during your reporting period and considered as safety issues under monitoring or regulatory authority request which was opened and closed, so in that case it will come under 15.1. But in 90% cases it will not be there. So did you are you able to get in which situation it will be there in 15.1?
So yeah, if like signal is evaluated or ongoing during your reporting period, or it is closed during your reporting period, it will be there in 15.1.
And if closed, then in 16.2 also.
Yes.
Any other question, people?
Amit, what is the answer for that question you asked in a group that left?
That is unlisted only, but I'll write it in the evening at night. You are correct. Whatever reason you have mentioned, all are correct. So it is unlisted
Okay.
only. Any other question, people? Yes, Irfan, tell me. Irfan, you have a question? Irfan, we are not able to hear you. Hmm. Let me mute him then. Yeah, muted. So people will start with our next session now, or still any doubt? Someone is asking, this is batch one session. Yes, this is batch one session. You people are just included because we are discussing sales today, which is independent of the previous section. You don't need to have any background. This will be a completely scratch session. So that's why you people are invited. But you will have these recordings and everything when your batch starts. So don't worry about it. Hello, then I'm presenting my screen and we'll start with sales and exposure section. Just let me know once you are able to see the screen. Okay. Where is that screen? Are you able to see my screen?
Yes.
Okay. So now today we will discuss about section 5. Okay. So can you tell me, anybody, what is section 5 all about? Anybody? Section 5 people. What is section 5 all about? Any inputs is appreciated.
What is the total exposure to our drug?
Not sales exposure, cumulative and interval.
Okay. Only sales exposure?
Clinical trial exposure.
We can expose
How many patients are
in
there?
clinical trial and one is in like in during reporting interval sales data calculation.
Okay. So only sales exposure you mean to say, whether it is from clinical trials or post-marketing, right?
Yes.
There is a section on other post-authorization use also. Have you heard about it? 5.2.3.
Off-label use, drug abuse, overdose may be included.
Correct.
Capital uses. Yeah.
Correct. All these will be included in 5.2.3 under other post-authorization use. Now, before moving forward, for sales exposure, like from either from clinical trials or from post-marketing or the other post-authorization use, can anybody tell me what is the significance of this? Why do we calculate the sales? It is a commercial part. Why do we need to calculate this sales? What is the use in aggregate then?
It helps to know that how many patients
Because
have
of the
been exposed
medicine.
to our drug.
Okay. Any other thing?
To calculate that how many number of patients or subjects are exposed and how many number of patients or subjects experience the event.
Correct. In this section, how many were exposed? You won't know how many were having the events. Okay. So in this section, you are correct that how many patients are exposed. So both of you answered correctly that we use this calculation to see how much our drug was exposed during the current reporting interval as well as the cumulative. Or always remember, sorry, this is the section in which we present both cumulative as well as interval. For exposure means from clinical trial exposure and from marketing exposure, not for 5.2.3 other post authorization. That will be interval information only. But for sales calculation, whether it is from clinical trial exposure or from the this one post marketing exposure, we will present both cumulative as well as interval exposure, right? Now, why do we calculate this exposure? So basic answer is, if somebody asks you like, what is the significance? Why do we calculate the exposure? Why is it required? So what happens? In RSI, you have seen the frequency of the events or the ADRs, you can say. How is the frequency? Like very common, rare, very rare. You have frequency, like suppose if it is very common, then in 10,000 patients, if exposed, you have that data, na? 100 in 10,000, 10 in 1,000, 1 in 1,000. You have that data, right? The definition for the frequency, common, very common.
In 4.8, hello Amit. Can anybody else hear Amit?
No,
No, I think their network is.
Hello guys, can you hear me now?
Yes, yes.
Yeah, actually the net connection was lost due to power cut. So I've connected again through mobile. Just let me know if it is. Now you can see my screen, right?
Yes.
Okay, so we were discussing that we have that frequency 1 in 1000, 10 in 1000, 10 in 10,000. Like that, we have a frequency, right? So what you have in denominator, that is exposure. That if 10,000 patients were exposed, 10 patients had those events, right? Are you getting it? From the
In
RSI, I'm
section
talking about the
4.8.
frequency, rare. Correct. In 4.8, you have frequency for the
Yes, yes.
ADRs. Like, so that's why we calculate the exposure in our aggregate reports to see whether the frequency is increasing or it is means it is constant or below the desired level, whatever is mentioned in the RSI. Correct? So why do we see whether the frequency is increasing? Because you remember, if the frequency is also increasing, then it is also a safety finding for us, and we have to provide the analysis and report it, right? Am I correct? Are you getting people?
Yes.
Are you able to correlate? Others, are you able to correlate why we are calculating exposure? Any doubt, people? See, calculation is not a big part, but why it is done and all, what is the significance? These are the main thing that you should understand. Others, yes, no, maybe. Give me a heads up so that I can move forward.
Sir, can you
Yeah.
tell the significance? Repeat the significance.
Okay. So see, if you have to calculate the frequency of your event or your risk, suppose there is 5 risk in your section 16.1, which you have to present an analysis in 16.3. Now, suppose I am having a risk of thrombocytopenia and its frequency is rare. Okay. And what is the definition of rare? Can you tell me, anybody? What is the definition of rare provided in RSI? How many events in how many patients exposed?
1 in 1000.
Anybody? I need answer from me. Please see into the RSI, any RSI if you have open, and let me know what is the frequency for that. Meanwhile, I'll open the Word document for us. See people and let me know. I think you people are sleeping. You are dependent now on a recording, I believe, nowadays. Speak up, people. Come on, tell me.
Yes, Amit sir, we are checking it.
Check and let me know. Until and unless you people speak up, I won't move forward. I need answer from you guys.
1 in 10,000 rare.
Okay.
Between 1 in 1,000 to 1 in 10,000.
No, this much should not be rare range. It would be 1 in 10,000.
Okay.
See clearly and let me know.
Greater than 1 in
Hmm.
10,000 and less than 1 in 1,000.
Correct. Rare frequency we are talking. So what is number? Frequency is number of events, put it events simply, upon exposure in patients, right? So now for rare, you mentioned what is the frequency that you are mentioning for rare? 1 in
Greater than
10,000,
1 in,
right?
yes, 10,000, and less than 1 in 1,000.
Great. 1 in 10,000.
Sir, I have ping one table. Is it correct from that chat?
Tell me, don't ping because I'm currently looking here.
It is less than 1,000 in 10,000 in one.
Oh,
Correct, it is whatever you have written.
oh, okay. So now suppose we were discussing about thrombocytopenia. Okay. So now for thrombocytopenia, your frequency is mentioned as it can be greater than 1 if the patients exposed are 10,000. But if only 1,000 patients are exposed, then it should be less than 1. Okay. Did you get the meaning of this? So now suppose you calculated the sales and your sales represent a data during your reporting interval that there were 10,000 patients exposed and thrombocytopenia cases when you picked from the line listing, there were 1 case of thrombocytopenia. Okay. So in this case, when the patient was exposed is 10,000 and event was 1 or maybe 2, that's also fine. So in this case, the frequency is not increasing. It is under the definition of rare frequency. Are you getting it? It is in the range of rare frequency, right?
Yes.
Yes,
So
yes.
in this case, there is no increase in frequency and whatever information is known to you, it is in line with that information. So you no need to provide any further analysis for that event frequency. You can say straight away that there is no change in frequency. Did you get the significance of this exposure section, where it is used? How is it used in the PBRER?
Yes.
I didn't discuss the frequency in detail while discussing for risk, that because I have not discussed sales. That's why now I am discussing how we say that our frequency is not increased. So this is the reason how can we say that our frequency is not increased. And you see, you have to do it for each risk. Like the exposure will be constant in interval. If you got the exposure of 10,000 patient treatment days or 10,000 patient treatment years, whatever is there. So the exposure is constant and the number of events will change for each race. So in that lieu, you have to compare, and if there is any change, then you need to, means if there is increase in the frequency, then you need to include into the your analysis as to what do you think, why there is increase in frequency. And if it is not there, then you can write a blanket statement that there was no change in frequency. Mainly we do it for important identified risk. You remember I told in important identified risks what we analyze? We analyze if there is any change in severity, if there is any change in frequency, right? Correct, people? Shall I move forward? Did you know what is the significant difference or what is the significance of exposure calculation? Any other doubt on this? So let's go to calculation, right? How do we calculate? Shall I go?
Yes.
Yes.
So, Amit, one question.
Tell me.
So, like denominator, we need to calculate from the clinical and post exposure, like how many patients are exposed.
Yeah. So actually what happens, you have to take from the post marketing basically.
Okay.
Because if your drug is into the post marketing phase, then you have to take it from post marketing.
Okay. Okay. Yes.
Okay, because in 16.3, whatever information you are presenting,
Yeah, yeah,
the
it's from
risks which you are
post-marketing.
taking, the events, that is from post-marketing.
Okay.
Any other doubt, Vibhul? So I think I should move on now, right? Okay. So see this Excel. Can you see this Excel and make out something from this? Anything your people are able to make out from it?
Yeah, I think it's a formulation wise data for the sales data. The total unit are sale from the department
Correct.
and the
So
region
see
wise.
what happens. Yeah, so I'll tell you. Just don't confuse yourself. So here you will get the information like suppose they will give you, let's take small drug name. Can you tell me any small drug name? CTZ, let's keep it as cetirizine. Okay, CTZ. Okay, this is cetirizine. Then they will say that 1 in 10 tabs and 5 mg. Okay, same way they can say that 1 in 10 tabs 10 mg. Okay, similarly they can say that let's say 20 mg. Let's keep these three strings. So what you can make up from this information? What do you get? What do you understand by this information? Cetirizine 1 in 10 tablets, 5 mg. You can make it 20. And let's keep it. Tell me, guys, come on. 5. Chalo, tell me. These three columns, formulation, quantity, and strength, will be filled from this information that I have currently populated. So tell me, what should I put in formulation?
It will be tablet.
What should I put in? Tab. Correct. And it is the same. So let's keep it as capsule. CAP. Okay. Chalo. Second tab. Tab again, right?
Yes.
Third.
Capsule.
Third. Cap. Correct. Now in quantity, what should I put in first?
10.
10. Why? One pack is having 10 tablets, right?
Yeah.
Second,
20.
20. Third,
5.
third, 5. Now in strength, what should I put in first?
5 mg.
5. Simple 5. Strength in mg you can write here. It will be, you will write here as mg. Okay, so 5. Next,
10.
10. Next,
20.
20. Correct. Now, what information should I give here? What should be the DDD? There are two types of things that go here, either DDD or RDD. So tell me what information I should put here. What should be the DDD? Will it be 5 mg? Will it be 5 mg, guys? Yes, no, maybe.
No.
Why?
Amit, what is DDD and RDDD?
DDD is defined
That
daily
one
dose,
is
RDD is recommended daily dose.
yes,
Yes, sir, I
that
think
one,
it
I
is
think that one is the reference we have to take for that.
Amit sir, I
Which
think
difference? Chalo, don't be confused because this is a new concept. So I'll teach you today. What is DDD? What is RDD? Okay. Let's come to our this. Now, what is DDD? Okay. So it is defined daily dose. Okay. And what is RDD? It is recommended daily dose. Okay. So now when we take DDD and when we take RDD. So for DDD, this is a defined daily dose and it is a standard one. So you can take it from a site which is called WHO DDD website. So let's go to Google one. Now I will put here WHO DDD. See all the steps that I'm doing, huh? WHO DDD. DDD. Chalo, let's open this. If we can find this is Uppsala. Sorry. Net is slow because I have connected it through. See, this side you can see ATC DDD index. So you have to go to this side. Okay. Now we are on this ATC DDD methodology. We have to click this DDD. Okay. This one and put here cetirizine. Where it has gone. Okay, index is here. Index, you have to come to index. Okay. So put here cetirizine. Okay. So we got cetirizine. Now there are two cetirizines. This is one is S and one is R. So this is the ATC number. So if you are working for your molecule and it is approved, so you will be having ATC number. You can find it from your previous PBRER or the tracker, whichever is there. So ATC number. So suppose our ATC number is this one. Okay. So we will go to this one. Now your drug R06 is for antihistamines and R06 is again for antihistamines for you. Okay. So now can you see here means this is the code which we clicked earlier. So that is the ATC code. And now for these, there are two R06, R06A. Both are for antihistamine use. Okay. So no issues with that. So DDD we will have to take same. So here you can see DDD. Can you see DDD here?
yes.
Or maybe yes, right? So you will take this DDD 10 mg and it will be constant throughout for the formulation. Okay. If the formulation is changing, then only you have to check whether there is some different DDD. But if it is tablet or capsule, if the same is provided, then you will take it as 10. So now were you able to find how do we get that DDD, defined daily dose,
Amit, I have
right?
one question here. Mm,
Yes, yes, yes.
yeah, that DDD is that reference, whatever here we are giving, that one is applicable for each and every
Hmm.
drug, or is there any exception here? Uh,
Yeah.
like,
So what will happen?
yes, yes.
Hmm. So what will happen like when you are working mostly on established products or generics and all those things. So this will be the reference means the reference site, and we refer this site also in our reference section of the PBER also. Okay. Now suppose if you are working on some innovator molecule, then their RSI will itself state that what is the defined daily dose. So you can take from there. Okay. And the next concept of RDD I am still about to discuss. So first let me
Okay.
know whether
Okay.
now it is clear that how do you get this DDD.
Yes, yes.
Okay. So now let's come to the next concept. RDD. Now suppose it might happen that you go to the WHO DDD side and there is no DDD provided. It happens. It happens usually. Almost in 20-25% cases it happens. So in that case, comes the concept of RDD, recommended daily dose. Okay. So in this case, what you will do, you will go to your RSI. For DDD also, you can go to your RSI. If it is provided there, then you can take DDD directly from there. But if it is not there, then you can take the DDD from that reference site. Mostly we take from reference site. Now, if we don't find DDD in on reference site, then we go to our RSI to check what is the recommended daily dose there. So in doses section of RSI, if any RSI is open, anyone can present, or just I'm telling you that in the doses section of that RSI, you will find as to what dose is recommended. Like suppose there would be a molecule, suppose there is some anti-neoplastic molecule or anti-cancerous drug. So they have cyclic therapy. So they will say that give 50 mg IV infusion and give it for 3 days in a month. Okay. Did you get what I'm saying? 50 mg IV infusion at one time for maximum 3 times in a month. Okay. So what do you think? What should be the RDD here? What should be the RDD as per you? So see, what is the dose that we can give at one time? That is 50, right? How many times in a month we can give? Into
3 times.
people. 3 is equal to 150 per 30 days. Am I correct? Am I correct, people?
Yes.
Yes.
The calculation is at 150 per 30 days. Now, since what is the definition? What does this say? RDD, recommended daily dose. Remember the fact, daily, right? And what you have calculated is for 30 days. So for one day, how will it be? We will have to people.
5 it is.
We will have to 5 mg per day. So this is your recommended daily dose. So in this case, RDD will be 5. So instead of 10 here, you will write 5 then, if you are not getting DDD from there. So this is how you will calculate recommended daily dose. Correct?
Yes.
These are the questions from the sales section which can be asked. So that's why I am explaining you with the example. Okay. Is it clear? How do we calculate DDD? How do we get DDD, or how do we calculate RDD?
Yes.
Yes,
Sir,
no, maybe.
Yes.
sir, can you give more example in RDD? How we calculate?
So that you can check. Na, if you have any doubt, you can get back to me. We have to cover many things. Na, if I keep on keeping example, then it will take time, right?
Okay, fine.
You check it from your own side. Don't assume. Just go to the RSIs, whatever SMPCs you can download. You can check what in the doses section, what is the dose provided, and in that you can come back with the examples. So if you have any doubt, I'll let you know as to how it is. Okay.
Yes.
Chalo.
So,
Let's move forward now.
hello, Amit sir.
Yes.
For recommended dose, it is that means in the RSI, it is mentioned for adult as well as for pediatric
Hmm.
patients.
Hmm.
So, in such condition, how we are going?
So in that case, what happens? See, when you calculate the sales now, this is the easy sale, means this is the most widely accepted sales that I have provided you based on the region and all. Then we have a stratification method, means sales can be stratified. It can be stratification means it can be bifurcated. Bifurcated based on the region, based on the age, based on the gender, whatever is your requirement as decided by the client, you will have to do that. Now what happens? Suppose if you are stratifying the sales data based on age, so in that case, you will get the sales information from the sales team based on age bifurcation. Like they will tell you that in adults, for adults, these many things were commercialized or sent into the market. For pediatrics, this one. So for adults, you will take what is the recommended daily dose for those adults. You will calculate for adults separately, and then for pediatrics, you will keep their recommended daily dose and you will calculate it separately. Did you get it the point?
Yes, yes. So,
Okay. So
that means, yes, yes, go ahead.
yeah, tell me.
So,
Any other
for
doubt?
some of the drugs, that means recommended daily dose is mentioned. Like that means I am referring one SMPC, and it is mentioned
Hmm.
that recommended dose for that particular drug is 200 mg daily, and occasionally up to
Hmm.
300 mg daily have been used in severely that means severely depressed patient. So.
Hmm.
That means how we are. That means here also. That means we are taking common recommended daily dose, or we are that means taking on that means on the basis of average like that.
Yeah. So when this type of scenario is there, you will take the 200 mg. That is the commonly recommended dose. Until and unless your means there is some health authority mandation or MAH has decided that they will database the sales, whatever sales are being done for those severely chronically ill patient, you can say, or the severe depressed patients. If they are giving the sales information stratified from them, means you have a different sales information for those patients who are having 300 mg, then only you will calculate by that indication. Okay. But if in case you don't have sales from them, then you can calculate from 200 mg. Okay.
Okay, okay.
And one more thing I'll let you know, because you are having doubt, because you are thinking that it should be exact. While providing the sales information, we initially we write the method as to how we have calculated or how we have reached to this calculation. And in that we write that whatever sales is presented here is an approximate estimate. We don't say that it is an exact estimate. This is an approximate estimate. Why approximated? Because whatever concerns right now you are raising, due to those, we put it as approximate estimate. Okay.
Yes.
Sure. Any other question?
No, I don't.
Okay. So I believe nobody else has other questions, so I'll move forward, right? Chalo. Then this section, this column, units. Okay. Now can you see here? I have stratified the data based on region, because in all the PBRs you have to at least present this stratification, at least region-wise bifurcation. So this is Europe, this is US, this is Canada, this is other row countries other than Europe, US, and Canada. So this is how we get the data in our sales sheet. Okay. They will give us the data differentiated by European region, US region, Canada region, whatever is our requirement, they will give the data according to that. Now, in the sales sheet, apart from this, what information we get in sales sheet, this particulars will be there. This section, this column will be there in sales sheet. This region will be there in sales sheet. And now this units will be there in sales sheet. Suppose they have mentioned 2,550 here. Suppose 3,000 here. So this data will be provided by them. Okay. So did you get the point? What you get in sales sheet? Sales sheet, we have this section. It could be particulars, it could be description, whatever is the heading that they will provide. So this column is provided by the sales team, and this column is provided by the sales team. both the columns which I have highlighted right now. And the region. Okay. Okay, people. Shall I move forward?
Yes, yes.
Okay. Now, what we need to calculate. These are the two columns that we have to calculate. Okay. So, total units sold. What do you think? How can we calculate total units sold? We can write this as units sold. Any guess? Now, whatever we will do, we have the data from there only we have to do. So, what formula should we put here to calculate this total units sold? Tell me, people. How can we calculate total units?
Sold during the reporting period.
Reporting period is fine. This is either reporting period, cumulative period. The same you have to follow. No reporting period or no cumulative period. Because whatever data they will provide you cumulative data as well as interval data. So from interval sheet, you will take the data for interval. This unit sold you will put from interval sheet, and this cumulative from cumulative sheet you will put unit sold for cumulative sheet. So let's suppose that we are doing it for reporting interval. So, but I'm asking like, how can we calculate this total units sold from whatever data we have currently that is shown to you in the, what should we do? How can we
With
get this total
quantity,
units?
with quantity or with DDD, with the help of DDD or quantity.
Yeah. So for total units sold, we will calculate it with quantity. Why? See, this is the pack size that they have given. See, you can see one pack into 10 tablets. So 2550 packs are sold. Okay. They are not giving you tablets. They are giving you packs. How many packets were sold? Right?
Yes.
Correct, people? So now how we will get the total units? So is equal to this packets
Units sold divided
into,
by 10.
no, into quantity, right?
Quantity.
How many quantity in each packet? So 10. So this is the total units sold. Okay. Okay, people. Any doubt? Tell me. Am I correct?
Yes.
Right?
I think it must be, it should be total units or total quantity sold.
Total units. It is here units sold. So you can say total quantity. No problem. Let's make it quantity. So it would be more clear. Total quantity sold. Correct. Chalo. Now it's clear, right?
Yes.
Now clear? Next calculation. What is this PTD? You can say it here as patient exposure in PTD. PTD means patient treatment days. Did you get it? PTD means patient treatment days. Guys, is it clear? Any confusion?
Yes, clear.
Clear, right? It is patient treatment days. Why we are taking it in days? It can be patient treatment years also. People can confuse you. What is the difference between PTD and what is the difference between PTY? Patient treatment years. Why we have taken it in days here, not in years? Any idea?
It's depend on drug to drug, no? Some drug we are taking, I mean, days in monthly or some in years.
Okay, see. Here we are calculating in patient treatment days because see this column. What does it say? It says defined daily dose or recommended daily dose. This is daily dose that we have taken, right? Right?
Yes.
Correct?
Yeah.
It is daily dose. That's why we are taking it in days. If it was yearly dose, defined daily dose, instead of that, it was defined yearly dose. Then we could have taken in years. We could have presented directly here patient treatment years. But eventually what will happen once we calculate this in PTD, and if the exposure is huge, so then we have to convert it into patient treatment years. I'll come to that part later on. But why we have taken days here, that is clear to you, right? We are taking defined daily dose. That's why we are taking it, calculating it here as patient treatment days. Correct?
Yes, yes.
Okay. So now there is a formula to calculate this patient treatment days. And this will be from these data, whatever is present here. So what do you think? What should we do to get it in patient treatment days? What is this? What is this, which I have highlighted currently?
Total quantity.
Total quantity. Total quantity of tablets sold. Okay, correct. So did the patients who consume, so shall we convert it to mg? What do you think? In milligrams, what is the dosage that we are using? Shall we convert it into mg? Because it is the tablets that are sold, right? But
Yes.
what was the total mg sold? Are we still aware of that? No, right?
Mhm.
So we should convert it to mg. So how can we convert it to mg? One tablet is equal to how many mg
10 mg.
5.
in this section?
5.
5. So you will multiply it by 5. So you will get total mg sold till here. Okay. Total mg sold. Still you have not reached to PTD. What we have got? Total mg. If we multiply it by strength is equal to total quantity. Just a minute. Is equal to total quantity into what is
10.
our strength of one tablet, and
10
then we will divide by till here
1,000.
G3 in
Okay.
till G3 into D3 you have got total mg sold. Now you have to
Mhm.
divide by per day defined daily dose or the RDD. So what is your per day defined dose? That is
10.
here,
10.
right? So when you click, you will get the patient treatment days. Correct? Did you get the formula? Why we use this formula? People, yes, no, maybe,
Yes.
confused, need to repeat. Tell me, people, I need an answer from everybody. Otherwise, I'll think that everybody, it is now understood by everybody, correct?
Yes, Amit.
Okay, then. So how we calculated? We saw that total quantity sold is equal to number of packets sold into quantity. So it gives us the number of tablets. Now to reach the PTD, what we did? We multiplied the total quantity of tablets sold by strength of each tablet. So it gives us the total mg sold. And then when we have divided it by defined daily dose, it gave us the patient treatment days. So what is the meaning of this patient treatment days? What do you understand by this term patient treatment days? Now you have got, see, there were 2550 units sold. You changed it to 25,500 tablets sold. And now you are changing it to patient treatment days, 12,750. What is the meaning of this 12,750? What do you understand by this patient treatment days?
That is the cumulative
So,
data.
not the cumulative data. Don't go in that cumulative or interval. We are discussing here interval data only. This is the interval data. Okay. So is it, just tell me, what is your thought? Is it the number of patients that were exposed during the reporting interval? Or is it the number of mg drugs which was distributed among the number of patients? What do you think? What could be this
I think first
figure?
option.
Number of patients exposed?
Yes.
Hmm. So this is
Um,
not the number of patients exposed. Okay.
no, no.
So what happens, since you have calculated everything based on dose and all those things, you never used a number of patient. You used a patient can take this amount of maximum daily dose. So everything is based on dose here, right? We are calculating the dose. Everything is based on dose. There is no number of patient. It might happen that two patients use the same dose on same day. So we are not calculating number of patients here. What we are saying here that the total mg of this 12,750 mg of drug was distributed into the market having a strength of 5 mg in our reporting interval, which was taken by the patients. Because we have a control over doses that we are sending into the market. We don't have control on how many patients are taking those. Like suppose if it was 20 mg a day, so it might happen that 3 patients have taken 5 mg tables, four patients have taken 5 mg drug. So we can't calculate the number of patients, right? But we can calculate how our drug was exposed, means how much of our drug was exposed into the market. Did you get the concept? This is not number of patients. This is number of mg of medication or number of doses that were sold into the market. It might have been taken by 100 patients, might have been taken by 500 patients, no. But this is the exposure of your drug into the market. Correct? Correct, people?
Yes, yes, yeah.
Yes.
Okay. So now what we do, in the same way, this is the small sheet that I'm showing, but it is a large sheet when we calculate some, the rows will go up to 1,000, 2,000. So in end, what we do, you can sum of all these things. Okay. So this is 11,004, 1,14,715 total mg, whether it was 5 mg, 10 mg, whatever is there. Okay. Now suppose if this data is too huge, so what we do, we convert it to patient treatment years. So how we can convert to patient treatment years? It's simple. Take this total amount divided by 365 per year. How many days? 365. So it will give you patient treatment years. Okay. PTY, right? Correct, people? Did you get the information? How do you get patient treatment years?
Yes.
Dividing by 365, right? Okay. In the same manner, you will take the sheet of US, whatever information about US is provided, you will put the units here and then you will calculate here. In the same way for Canada, in the same way for other than these three countries. Or if somebody says to stratify it for Japan also, then you will put Japan separately. Some might say for some other region, so you will put that region also. There will be in your sales data. And then whatever is remaining, they will come under row countries. Am I clear? How do we calculate this sales? Am I clear, people?
Yes.
Yes.
Okay. Okay. Then. So this is the sales calculation done. Now, how do we present in report? So the same way as you have calculated, you will prepare a table here. I'll prepare in front of you so that you table here. Suppose we are having 4 regions, right? And 2 formulations. 1, 2, 4 regions. This much. Anything else? I don't think so. Let's put it like this. So here you will put all indications. No need to remember these things, but just I'm showing you how we present this data in the sheet report. All indications, then you will keep it as formulation. There are 2 formulations, so we'll keep it at, then we will come to region. Okay. So formulation, we have 2. One is tablets, then one is capsules. How many formulation you have? You can present that. Then region, you will go to EU, US, Canada, and then row. Okay. You can merge it. Merge cells. You can merge this also. These are all formatting that can be done at any time. So just I'm leaving it. So suppose tablets. So we will present in patient treatment years. If we are getting in patient treatment years, then we can present in patient treatment years, or means because patient treatment years, why we are calculating? Because the data is used. So we are presenting in patient treatment years. And before writing this table, we will give a methods here. There will be methodology used. Okay. So that is a standard statement. No need to worry about, you will get when you are writing methodology. So this methodology will say as to what DDD you have taken, what was the formula used, and all those things that we used in calculation sheet. So from there, like suppose if you are asking me to put a tablet, so where you can get this in formulation, just put a filter here. Why the filter is not applied? Performance logging started. What is it? Why it is not giving that? Hmm. Oh, sorry. So I'm not getting this filter option. So you will get the filter. So you will put a filter of tablets. So these two columns will be there. So for example, we can take here, what is the sum of these two columns? So this is 72,750. You will write here 72750. Then for capsules, you will get, so capsules you have this 42,000. So you will put 42,000. Then for EU region, so you have calculated only for EU. So we have 11,4750, 11,4750 for EU. In the same way, you will calculate everything. And then here you will give total. Total means the total of these two in one column, then total of all these four regions in this. Okay, did you get how we present this data? People, tell me, did you understand how do we present this data? So this will always be, since we have kept it is here, this one. So this is the, since we have calculated only for EU, so the total is same. Otherwise, the total could have been the sum of all these four. Right? Any doubt? Till here for sales calculation, any doubt? This is post-marketing sales calculation that we are doing. Okay? People, yes, no, maybe.
Yes, sir.
Okay.
Yes.
Then. So now I think that you people are okay. How do we calculate the sales? Okay. So now for clinical trial sales, what happens? For clinical trial sales, you don't have to do anything. The clinical trial team provide you the exposure. That exposure is from DIBD till the DLP. So it will have both, they will give you the interval also and they will give you the cumulative. In clinical trial, the cumulative is from DIBD, Development International Birth Date, and for post-marketing, it is from IBD, International Birth Date. That is the difference. In clinical trials, they will provide you data bifurcated, how many patients were exposed, how many subjects were exposed to placebo, how many subjects were exposed to treatment drug, how many subjects were exposed to comparator drug. So in that way, they will provide you these data. You don't have to do anything. You have to take the data and simply paste it into the PBRER. Now both the sections, the clinical trial exposure and post-marketing exposure, are complete. Correct? Any doubt till here?
Amit sir, can you repeat this clinical trial data?
Yeah. So what will happen? You will request that exposure data to clinical trial team. So for like post-marketing, you will expose the sales data from the sales team. So they will give you the sales team, the column that units told we were asking about, and the description or the particulars. So that will come from sales team. And from the clinical trials team, they will give you the exact exposure, because we don't require defined daily days and all those things, because that is a protocol controlled study. So they will directly give you the number of patient subjects that were exposed. So there no patient treatment here, no patient treatment is nothing you have to do. No, you are not required to do any calculation there. They will give you the number of subjects exposed directly. So you will take the data directly from there, present it in the table. The table format same as to the table format will say that number of subjects exposed to placebo. So suppose 1000. Number of subjects exposed to treatment arm. So suppose 5000. Then number of subjects exposed to comparator arm. So suppose 500. So this way you will present the data there. So that will be a directly drop-in section from the whatever the clinical trial team is providing you. Okay. No need to do any calculation there. That is a direct section.
So you mentioned about from DIBD to OCT.
From DIBD to DLP. Yeah. So if it is cumulative, then you will calculate from, you will give a two type of data. One is cumulative, one is interval. So cumulative will be DIBD, that is Development International Birthday. We discuss what is DIBD, right? From
Yes, yes,
DIBD
yes.
to DLP.
Mhm.
So that will give you the DIBD to DLP, that is cumulative. And for interval, it will be the reporting period which you are talking about. So this will be the exposure from clinical trials. It will not include post-marketing. It is separate. It is a totally separate thing. Okay. This will not include your post-marketing, whether it is cumulative clinical trial exposure or whether it is interval exposure. So it will purely be into the clinical trials. How many subjects were exposed into the clinical trials? So that is a drop-in section coming from clinical trial team. For post-marketing, the cumulative will be from IBD to current date. Because when did your drug started to be exposed into the market? After the approval, that is IBD. So from IBD to DLP is cumulative post-marketing exposure. And for the current reporting interval is your interval exposure. Correct?
Yes, understood.
Okay. Any other question, people?
So Amit, just one query. So we can refer the previous PBR for the last DLP, and we can add on that last value with the current interval period.
No.
We are
Why?
receiving because
Yeah.
till last DLP we have the data, and whatever the sales data we receive for the current interval period. So can we refer the previous data and just add the current interval period?
For cumulative?
Yes.
No. Why? I'll tell you. What happens every time you will, when you are writing PBR, you will request a sales data from new sales data for the cumulative as well as interval. Why? What happens? Suppose in the last reporting period or in the last PBR, the cumulative sales was some 10 lakhs. Okay. Then what happened in your reporting period? It sometimes happens that few you can say shipments were cancelled and called back. There is some market recall, or due to some reason the tablets were returned back to the company. So there are returns also in that. So how will you manage those? So that's why we request fresh data every year while writing the paper, so that we can give the current information. You should have heard about product recall and everything, right?
Yes, right. Yeah.
So that is the reason we don't sum up from there.
Mm-hmm. Yeah, like some expired product also returned to that company.
There might be returns due to n number of reasons.
Right.
So that's why we request fresh data.
Yes, got it.
Any other question, people? So, okay. Let's move to then next section. What is the next section now? What is the next section, people? You told me in the beginning what is the next section. 5.2.3 other post authorization use, right? Right, people? So now, so people might ask you as to, and I have heard some people have been asked that question, that what is the difference between section 5 or specifically they are referring to section 5.2.3 and section 9.2, that is medication error. That is 5.2.3 is other post authorization use and 9.2 is medication error. So what do you people think? What is the difference between those?
It is off label and 9 point is medication intervention.
That is given, that is from the headings itself you can identify, but what is the difference? What is the basic difference between that? See, I'll tell you. The basic difference is in the medication error section, we are presenting the cases which was used for approved indication, right? In that medication error section, I discussed that whatever product used in unapproved indication or drug ineffective for unapproved indication or off-label use, if it is reported as a single event in that case, we will exclude it from medication error. Single event means there is no other medication error related PT in that. If these are the only PTs reported in that case, then we will exclude it, saying that cross-referring it to section 5.2.3. So what is the difference? In medication error, we take the cases which were used for approved indication or which were intended to be used for approved indication. But this 5.2.3, as it itself states, other post authorization use means, what do you mean by other post authorization use? So you are authorized to use it for suppose non-small cell lung cancer. But other than non-small cell lung cancer, where are you using it? How are you using it? So it is outside of your label. So that is the difference. This section 5.2.3 talks about what was the use of the drug in other than the approved indication or outside the label. Did you get the difference? What is the difference between these two?
Yes,
Is
sir.
it clear, people? Other
Yes,
post
sir.
authorizations use?
It's for unapproved. Like it is not.
Your voice is very freaky. Why you are not at home, I believe. So is it clear, people, right? Other post authorization use? What is the difference in this section from the medication error? Right? Yes, no, maybe. I think people are now mostly dependent on the recordings, I believe.
Can you please explain with example, like if it is like
Okay.
off-label use, patient, doctor are intentionally use, or it's like.
Mhm. I'll give an example, don't worry.
Mm.
Any other doubt? Okay, chalo. Now suppose minoxidil is your drug. Approved indication is hypertension, suppose. Okay. Now being used for baldness, as you know. So your approved indication is hypertension, but the drug is being used for baldness, right? And now when you get this case, you have coded it as off-label use or product use in unapproved indication or other same terms. Okay. So you have coded it as for off-label use, right? Right, people?
Yes, yes.
Okay. So if it is off-label use, then it will come under this section. If it was used for hypertension and no safety related issue was there, so would this case would have been pulled into your line listing in any of the search criterias? No, right? Because there was no safety issue related to it. Correct?
Yes,
It would
yes.
not have been databased. But since there was an event of off-label use, it was database and pulled into your line listing. So where should you present this case? So you will come to other post authorization use 5.2.3 to present this case. Did you get which type of case you present in off-label use? Is the difference clear now?
Yes.
Okay.
Hello,
So
Nick.
how do we Yeah, tell me.
Yeah, is it only with respect to indication?
Yeah, off-label use is used to
No.
in respect to indication, in respect to population, whatever is the use other than that mentioned in your RSI. I mentioned now outside of your label, whatever is the use practice or prescribing practice for that molecule, it will come under off-label. Did you get it? Like suppose if you say that your drug is approved to be used in adult patients only, and now it is being used in pediatric from the prescriber. So this is also off-label use, right?
Right.
It will also come under this section. Whatever is outside your label.
Yeah. Hello.
Yeah. Yeah.
Amit, misuse and abuse cases also will be here.
Yes, they will also be here. Drug abuse, drug misuse. That is they will form subsections. So first I am covering this off-label use, then we will cover drug abuse and misuse. Or you can keep it here itself, no problem, because there is no such differentiation in both. So, but there will be two subsections, remember. Drug abuse, misuse. What you can think of other terms? Drug abuse, misuse, overdose.
Intentional overdose.
Intentional overdose. So we will keep it for overdose right now. What else could be there? These are the things mainly. Chalo. Then now, what is off-label use? What is drug abuse? What is misuse? What is overdose? So all these are apart from your label. It is not mentioned in your label to use these things. So these are being used. Some drugs are there, like some cannabinoids are there. So alprazolam or so they have a tendency to be abused or misused also. Some drugs are used for you can say suicide purposes also. So that is misuse and overdose. So these type of cases which is outside of your label, all these will come under this other post authorization use. Okay. So now what we do, suppose we are doing off-label use, drug abuse, misuse. So we have search criteria like for drug abuse, misuse, we use broad SMQ of that drug abuse. And always we are using broad SMQ so that we are not missing on any of this. For off-label use, we have certain PTs only which will give you the off-label use, unapproved PTs or off-label use PTs. So you can use those combination of 4 or 5 PTs and you can get the cases. So this will be the search criteria for this. Okay. After that, when you get the cases, what you have to do, you have to look as suppose if it is an off label for indication. So you cannot blindly include it here. So you have to go there and see. Sometimes it might happen that ICSR people have done it incorrectly. They have incorrectly coded the off label use. So you have to go into the case narrative and see for what indication it has been used. Was that indication actually off label use? Then you will include this case. If it is not, then you can query back to ICSR and then you can move ahead as whatever the solution is there. Okay. So so you will differentiate the cases based on the off-label use, abuse, misuse, and present in the respective section. Now, what will happen for drug abuse, for all these things, whatever is there, so you will look specifically for the indication PTs, what indication they were used as, and that you have to present in the case, this section. Okay, like suppose if a drug was used for off-label use, you are presenting for off-label use section, you will see what were the indications. Suppose hypertension is the indication here, but minoxidil was used for baldness, it was used for some vomiting, it was used for diarrhea, it was used for something else also. So you will present the bifurcation of those indication PT. So you will write like we receive means the MAH retrieved suppose 100 cases of off-label use based on the search criteria that was used. Okay. Now you will write that of this 100 off-label use PTs, the most commonly reported or the reported indication PTs were this. So you will provide a table of that from as we used to do there. You will do a PT pivot and provide the table. So this table will be like the same way as we are doing for that one. अरे, चलो. Indication PTs, number of case reports or cases you can say, and then you will do. Suppose it is baldness, so suppose 50, and then suppose vomiting 25, suppose then you can say simple. I'm taking not too much. Yeah. This way you will present this all whatever PTs were reported as off-label use for your indication, and then you will see if there is any trend of being identified. The same way as you do for medication error. Okay. Now the process will follow the same. The if there is any trend, then you will go to each narrative and see if there is any specific pattern that is being observed. If yes, then you have to provide analysis of that pattern. Okay. And if there is any safety issues associated with, means some AEs are reported, then you will say that of these 100 PTs, 100 case reports of off-label use, there were these many PTs reported, out of which you can show how many were unlisted AEs, how many were listed AEs. For unlisted AEs, you can present the causality. While you are representing the causality for unlisted AEs, you have to say that, first of all, you will say that this, as per RSI, the approved indication is this. However, the drug was used in the off-label use for this. And after that, you can provide the ACE causality stating simply. Or if you don't provide causality here, then also it's fine, because we are looking at what is the indication PTs. Mainly, there won't be any AEs. Mostly we don't get, but if we get, then you can provide the causality. There is no hard and fast. Okay. If there are unlisted A's, then prefer to provide the causality here. The same way you will present the drug abuse, misuse, overdose cases also.
So Amit, this is about indication PTs. Then there is off-label for dose, frequency.
So
for
you will
more,
present accordingly. Now, first you will present for why it was for off-label, why it was considered as off-label. So if it is for indication PTs, then you will provide indication PTs. If it is for patient population, you will provide for patient population. So in that you will provide it will be hardly, means suppose if it is adult cases and you are getting exposed to pediatrics, so you can present here that off-label use due to special means uh, other than the means you can other population you can say. So in that you can provide that pediatric patients exposed, how many case reports were there. So you can provide if there was any safety findings, then you can present that safety findings. It is simple. You can bifurcate as to based on what was the off-label reason for that. Okay.
can you repeat the causality? How do we like assess?
For causality, if there is unlisted A is reported with the off-label use, so before writing, means when you are starting the causality assessment, at that time only, first statement will be that this was used for unapproved indication. What was the unapproved indication? You can say that, okay? Because you can write according to RSI, it is approved for this indication. However, in these case reports, it was used as is in unapproved indication as mentioned in above table, right? After that, you can write whatever causality you have got, all the 6, 7 terms, criteria that you are analyzing for each. Na? In that case, you can use those and write your causality. Mostly we try to identify the trend or pattern here, if there is anything. See, I will tell you what is the reason behind this. What happens? Why do we do this other post authorization use? It happens mainly in the innovator molecules or the molecules which are into the market from for the first 5 or 10 years, you can say at that time only. Like for this minoxidil example only, okay, it was approved for hypertension, and when the cases were being analyzed, it was found that it is widely used for baldness. So it is having a side effect of hair growth. So that's why people are using to grow their hairs and all those things. So this is an additional beneficial use of this minoxidil. So the MAH again studied it for hair growth, and they have launched the minoxidil hair oils and everything for baldness, got it approved. So this is basic reason we try to see what is the trend or pattern being observed, how we are using this, what is it some beneficial use that we are getting from this off-label use and all those things. And if it is drug abuse, misuse, overdose, then we need to have some minimization, risk minimization, because it can become a risk for us when the drug is being widely used as a abused or being misused or being taken as overdose to commit suicide and everything. So this is a risk for the drug. So it will be eventually turn into a risk for your molecule post signal evaluation, and then you will have to have a risk minimization plan for this. So in that case, you will restrict the use of this medication and all. So that's why we use this post authorization use section.
We are assessing causality in other safety section with event, like if it is a signal or risk. In this section, what causality we are assessing, like with indication we are assessing causality.
No, no, it's fine that it is being used for off-label use, but causality, why we are doing? Because we are getting some unlisted A's. So whether your drug is being used for off-label use, you are writing that statement. You are correctly stating in the statement that it is being used off-label use in this cases, and then the A's happen. But still, your drug was taken, and it is you have to show whether your drug was culprit, you can say, to cause this AE, whether it was used in off-label use, no problem. But you have to state that causality, whether you think that your drug was a causal culprit for this, or there were other confounders which could have caused apart from being used in off-label use. So that's why we use the causality. We do causality here.
So lack of efficacy event also we will choose present.
See, lack of efficacy event we don't present here if until and unless like you can get, suppose if it was used for off-label use. So some people might report that it didn't work for me. So it is obvious that it is not approved for that indication. So how it can work? So it can come under the PT section for you, that drug use, drug ineffective in unapproved indication. That could be a PT. Okay. So you can present that PT, no problem. But we don't assess the lack of efficacy cases. For PBER, if the lack of efficacy is from clinical trials, then only we assess those cases in that respective section where there is a section on lack of efficacy in controlled clinical trial. Apart from that, we don't assess lack of efficacy cases anywhere in PBER. Now, but nothing is a thumb rule. For Canada region, if you are preparing a PBER, so Canada Health Authority mandates you to assess the lack of efficacy cases. That is a specific criteria for Canada Health Authority. So if you are preparing a PBER for Canada region, then in section 9, after that 9.2 medication error, there is you can create a subsection of 9.3 lack of efficacy, and you can assess all the lack of efficacy cases in that section. But this is specific to Canada only. Some other region also might mandate, or it also might happen that there is some molecules which are used for life-threatening condition in which HA mandates you to monitor the lack of efficacy cases. So you can present those cases. Otherwise, no need to present lack of efficacy. Yes. Okay. Any other question, people?
Uh, Cells, regarding cells, we are doing it because we want to see the frequency if it is changing, right? So, uh, but in that RSI, we are seeing frequency like in 1 in 10,000 patients. But here we are presenting cells in form of mg, how much mg is exposed. So how do we calculate number of patients then?
No, that is not, you cannot say that that is not number of patients. That is means that is the exposure. In the RSI also, whatever you are getting that by denominator, that is denominator, that is the exposure only. If the drug is exposed into these many patients, so that is not number of patients exactly. You can't say that that is number of patients. That is the number of Why? That's why we use the term exposure here. We don't, otherwise we could have said directly that number of patients exposed, right? But we are saying exposure of the drug during cumulative interval. And in the same manner, that frequency is also presented there. For our easy reference, we say that it is 1 in 10,000 patients, but is actually the drug exposure in 10,000. So it could be either in means ideally it is in patient treatment years, that is the most widely accepted. So that whatever denominator is provided, you can directly correlate to the sales section. No need to worry that it is number of patients and don't get confused also. Just I told you for your knowledge, so that if somebody asks you that what is the patient treatment year, so that you can tell them easily. But nobody correlates like that. That is the same thing, both in RSI and both in here, whatever you are calculating in sales.
So that number denominator is the amount of drug in mg or
That is exposure. That is exposure. Not exactly. That is not exactly the mg also. So for the time being, I have told you, so forget that, or just keep in your mind, don't confuse it with yourself. That, see, sales is a approximate. That's why we are doing it in exposure term. You cannot say that that is a number of patients. That is a number of dosage or the mgs that are distributed among the patients. So don't confuse with that. Okay. Don't correlate it with number of patients, or there is nothing like that. It is simply, you have to keep in mind that that is the exposure of the drug. And if somebody asks you what is patient treatment years, first define them by how you calculate the patient treatment days and treatment years. And then if they still want to know something, if they are still insisting to know, then you can tell them that concept, that this is the number of mg of the drugs that was widely that was distributed during the reporting interval among the patients. So don't confuse with that.
so why we have divided it by DDD total mg.
So the total quantity that you got, how did you get that quantity?
So by unit into strength.
No, this total quantity, how did you got? How did you get this total quantity?
So how many
Units
units have
sold
been sold into strength of that?
No, it is number of tablets,
Yes, sorry, tablet.
quantity, right? Now, when you are taking this defined daily dose, so it means that your drug is intended to be used 10 mg in a day, right?
Yeah.
What is the concept of this DDD? That this is the maximum dose that a patient can take in one day, right? Right?
Yes.
So this is 10 mg. Now, what is your strength of the tablet?
This
Strength of
5.
tablet is 5 mg. So when these many tablets were used, so how many mg was used? How can you calculate the mg? See, for doing any calculation, you have to bring every number into a single unit, right? So here everything is in mg. So you have to bring this tablet also in mg. That's why then only you can have that formula applied, right? You can't apply one thing in mg, one in kg, like that you can't do. You have to convert everything into mg, right?
Yes.
Here. So this is total tablets. Now, how many mg of tablets? How will you calculate the number of mg of tablets?
Total quantity into strength.
Total quantity into strength. So this will give you mg. Okay. So this is total mg of tablet sold during this reporting interval. Okay. Now, how many mg was consumed by the patient. Means this total mg was distributed. So how many patients could have approximately consumed this? Right? Right?
Okay.
Suppose if you are having 5 apples at your home and you say that there are 3 members in the home and you say that one can consume one apple only in a day. So 3 apples are to be consumed in a day, right?
Yes.
So totally how many days that apple would last? Approximately 2 days,
Yes.
right? Approximately 2 days that apple will last. It will not happen that you can say that only 3 members ate or only 2 members ate, right? You can't count the number of people who ate that. But approximately it may last up till 2 days because you can say 3 per day. So 6 you are having. So it will last for 2 days. So you can calculate how many days it can last to maximum. Means minimum it should go for 2 days. After beyond that, it can go to 3 days, 4 days if someone is not eating, right? So you cannot count how many people ate that, but you can count as to how many days that apple would last based on your criteria, the defined daily dose or the recommended daily apple consumption that you have kept in your house, right? So in the same manner, when you have calculated the mg, then how many days that mg will last. So you can't calculate the patient, you can calculate how many mgs that tablet will last. So for that you have divided it by DDD. Got it?
So
Did you get
whatever
the
number
concept?
we got in
See,
DD,
can you see the
DD.
formula here? Can you see the formula
Mhm.
here?
Yes, yes.
Number of tablets multiplied by strength, it give you
Mhm.
total mg. Okay. Now per day you have said that you can consume 10 mg. So divided by 10, so it give you patient treatment days. So it give you the number of days that drug will last in, be it consumed by 100 patients, be it consumed by 10,000 patients, whatever is there. So that's why we have divided it by defined daily dose, because we have the concept, we have the data for that defined daily dose, right?
Yes, yes.
Am I clear? Hmm? Are you getting the point?
So this PTD number will convert it into year if it is huge data, right?
Yeah. So when we convert it into year, because patient treatment years PTY is most widely accepted, and since this is a small sheet, I have taken only 3 rows. So it will go to 1,000 rows. It will go to 500 rows. So the number of patient treatment days will be a huge number. So to reduce it, we have divided it by 365 to get the patient treatment year. Sometimes it happens that we can convert it to, if it is more data, like for generics, cetirizine, paracetamol, we get even huge number in PTY. So in that case, we convert it further to per 1 million patient. So whatever patient treatment years we are getting, we will divide it by 100,000 or 1 million, then we will get the million patient treatment years exposed. So if the data is used, then we can reduce it. And these are the things how we reduce it. And most widely acceptable is PTY, patient treatment years.
So this PTY is the same number we have in the RSI, that frequency wala.
You are confusing with the RSI. Why you are going to that RSI? How can you get the PTY in RSI? Tell me. RSI
So
is based on certain analysis. That is a cumulative, or you can say certain analysis. That is a set criteria for frequency. Okay, that 10,000 and 100,000, that is not changing for other molecules, or in every RSI you will find that. That 10,000, that is a standard the definition given for that exposure frequency.
so how we
Did
are
you get my
correlating
concept?
then? I mean, like how we are correlating the frequency with the exposure?
Okay, let me tell you. See, you have got 1 in 10,000. Okay. So this 10,000 is exposure. Don't confuse that it is number of patients or number of mgs sold. This is exposure simply. Okay.
Okay.
Did you get the point? Exposure. Now, when you are calculating in your sales data, you are calculating the exposure only, right?
Yes.
You're calculating the exposure. Suppose you got exposure is equal to 5,000 patient treatment years. Okay. This could be patient treatment years. This could be patient treatment days. This could be million patient treatment years. No problem with that. This is the standard. This is not patient treatment years. This is not million patient treatment years. This is the standard definition for each molecule for the frequency of rare, very common, common. So don't confuse with that. Okay. So now see, this is one thing. So what will happen? You got 5,000, you will compare your data around it. So this is the one data comparison that you can do from this one for frequency. There is another method I can tell you. I don't want to go put you people much into depth because you will confuse everything. If you will calculate by, suppose, there is another concept of reporting ratio. If you are confused with this, then you can use this. There are many things in sales, but I can't tell you everything because that will confuse you only.
No, I
Chalo.
just wanted to know what is the significance of this sales
So
section.
that's what I'm telling you. Just a minute, because this is the exposure section. That's what I'm telling you now, how you can calculate it otherwise also. If you are not going to, because you are all time confused with this 1 in 10,000 that is given in RSI. So let's forget the RSI. I just give you a simple example of RSI. Now let's go to some more easy data if you want to understand. Because everything, if you would like to know, then you will be confused. That's why I'm not explaining you those things. Chalo. There will be risk term or PT, you can say. Frequency comparison, I am telling you. Okay. Now, risk PT is there. Then number of risk events. Okay. Interval. Then cumulative. Chalo. Okay. Now risk PT, suppose thrombocytopenia. Total risk reported 10. Interval exposure. This is interval exposure, remember, right? Exposure. And this is cumulative exposure. So if you are not getting the concept of that RSI, then use this concept to calculate frequency. Now interval exposure is suppose 3 or 5,000. Cumulative exposure is suppose 40,000 for thrombocytopenia. Now let's see hypertension. Number of risks reported, suppose 15. Interval exposure will be same, 5,000. Right? Interval exposure will be same, right?
Yes, yes, it will be.
Am I correct? Cumulative will also be same, right? Only for 2 risks will be take then. Then write what is reporting ratio. Okay. So reporting ratio, what you can do here also. Chalo, for interval we will calculate and for this. What will be for interval? 10 upon 5,000, right?
Yes.
Is equal to how much? 0 point. Tell me. 0, 0. Tell me, guy.
2.
How would it be? 1,
Two.
right? 2. Is it correct? 0.002 or 0.02?
002.
Right. Here is equal to what will be?
10
What would be the
upon 40,000.
Why 10? Why 10?
So thrombocytopenia. Number of risk.
Number of risks. So it should be 10?
Yes.
No. We will add one more column here. If you are calculating it by interval, then it should be with interval. Then here you have to calculate the cumulative. So you will have to additionally download one more line listing for cumulative risk. Okay. So now suppose in cumulative, you are getting some was 250 cases. For this, you are getting some 550 cases. So now tell me what it will be here is equal to
Is equal to 250.
250 by
40,000.
40,000. What is the answer? Tell me.
0.00625.
Let's do it here. Do 250 by 40,000. What is 0.006? 0.006. Now tell me, is this greater than this? Is
No.
this data greater than this?
No.
No. So what is the reference inference from this?
That it
What is the
has
reference
not
from this?
increased.
It has not increased. So now you are clear from this method. In this
Yes,
method,
yes.
you have to have the line listing for interval to that you will have, but you will have to line listing for thrombocytopenia specifically for cumulative as to how many events were reported for thrombocytopenia during the cumulative also. And then you can use this method. So this is one of the methods that you can use in your aggregate reports to give the frequency. Okay. So now there is any confusion?
Okay. So for each identified risk, we have to do this.
Not only identified, for all the risks listed in 16.1, that is either important identified or important potential, not for missing information.
Yes. Okay.
For important identified and important potential. This is the way you can do the frequency analysis. Okay.
But can't we just refer to the RSI and say it is not increased?
But then you are confused. Ideally, first I told you now you can refer to the RSI and you can say, but you are confused with the patient treatment years, So you can use this method so that you don't have confusion and you didn't jumble up in the interview. You can use this reporting ratio concept for frequency. Okay. Ideally, you can use the one that I told you earlier. That is also fine. Nobody will question you. But in case if you are confused and you are
Amit sir, we can't hear you.
We
okay.
can't hear you.
Amit, could you
Hello.
please
Can
repeat? Yeah.
yeah. So I told you that the first I told you can hear me, right? I am getting some message on this.
Your voice is
You people can
breaking.
hear. Hello.
Yes, now audible.
Now it is clear.
Yeah. So I told you the first method that has to you can calculate that and based on the number of risk for the interval, you can see and you can say that there is no increase in the frequency from the RSI itself, from the very rare frequency. Suppose in RSI this is not provided, or suppose if you are confused as Charuk was confused, so you can use this reporting ratio method. This method was mainly this method is used in CARS, that is Canadian Authority reports, to calculate the reporting ratio and all. But you can use this method in PBRER also if you want to explain this in your that PBRER report as to whether frequency is increasing or not. So you can use this method of calculating from the reporting ratio, comparing the interval cumulative, and you can say that whether the frequency has increased or not. There is one more thing, that the people take interval exposure for the previous reporting period from that PBRER, and then they compare whether there was increase from previous reporting period or not, whether there is increase from cumulative exposure or not. So there are many things that can be done to calculate the frequency, but let's keep it simple because you are not doing. So don't go into much complex things. But this is easier. You can use this. But apart from that, or if you say from the RSI, then also that it's fine. Don't worry about it. Okay.
All data come from sales team, or we have to calculate all these things?
You people are not paying attention in the training. What I told you in the beginning, this column and this column, this comes from the sales team. Rest all we have to populate and calculate.
Okay.
Also, Amit, in RSI, the frequency will be the same. It will not update.
No,
So, yeah.
it will not update. That is not the case. Suppose if the frequency is increasing and you are doing a signal on that frequency itself, and you find that now this frequency has increased, so you will update your RSI in that case, na? Changing its frequency. But ideally, it doesn't update.
Yes, that's what I'm saying. Like, if we are assessing a risk, whether its frequency change or not, we are doing it from RSI only, right?
RSI, I told you a reference that you can use to say about your frequency whether increased or not. But if you are presenting it in PBRER, then the formula which I told you right now, you will use that, na? You can't say the RSI there, right? You will use
That's
this
what I'm saying. Like,
formula when
this
you are
formula will
presenting.
be used.
Yeah, if you are presenting it in PBRER report specifically. And in this formula, you can also use, but mostly 98% people don't use. You can use one more column of interval exposure for last PBRER reporting period, so that you can compare the frequency from last reporting interval to current reporting interval, then from current reporting interval to cumulative. So you can say that there was no increase from last also, and there is no increase from cumulative also. Okay.
Yes.
So this information will go in 16.3.
16.3.
For characterization, also we can use this formula.
No. For characterization, you have to refer RSI. That is a standard that has been already reported to your health authority. Either RSI or your clinical trial CSR for clinical trials frequency. Any other question, people?
So Amit,
No questions?
yeah, one question.
Yeah, tell me.
So like if a company having the
Shall I
some
stop just a minute? Shall I stop sharing now?
yes.
Okay, go ahead.
Yeah, so company having some business partner, so we are requesting their data also for the exposure purpose.
Yes, if they are the marketing authorization holder, then we will request their data also and include it in our sales.
Okay.
Because they would be marketing authorization for certain region only. So for
Right,
that
right.
region, their data will come.
Yes. Okay.
So wherever there is agreement for use of our drug, we will collect data from there also.
agreement means they should be business partner. Otherwise, your sales team will have all those data wherever you are selling your drug.
MH. Yes.
Wherever you are
Suppose,
MH,
that drug which is used in clinical trials, which is not sponsored by or supported by our organization. So who will provide this data?
so when we are not sponsoring, then how we are responsible? We no need to present that data. We are not responsible for that exposure if we are not sponsoring anything. If something is there that the investigator has initiated, but we are sponsoring the medicine to them, then we will provide that in the investigator sponsored clinical trials. But if we are not providing data, they are procuring from whatever source they have. So we are not responsible to calculate, because suppose let's take an example. An investigator is doing a clinical trial on his own. So he could have procured the drug from some distributor or somewhere, right? So how that distributor could have got your drug? It should have been sold by your company only, right?
Yes.
So when your sales team is providing the data from SAP, so they have that data entered, whatever sales was done to that distributor who gave the drug to investigator, right?
Right.
So that data is under post marketing for us.
Yes.
So we can calculate only those things on which we have control. How it is being used into the market, we don't know. Okay. So if there is something from that market, people doing something on their own or that, and we are not monitoring it, then we are not responsible. How can we trace that things? So we provide only the data that is available with us.
But I mean, there are trials, clinical trials I'm talking about, where other companies use our drug as a comparator.
So they need to provide
To,
as a comparator, na there. See, suppose if you are not having a control on the sale, means distribution of their data, like suppose if a company is doing a clinical trial, okay, and in that clinical trial, there is no role of yours as a company. You are not giving them your molecule. They are procuring molecule from somewhere else. They are using it as comparator. So your molecule is widely available in market. So they are procuring for market that data. So in their PBRER reports, the MH who is using your drug as comparator, they will present that data in their report. That's why we are also presenting the comparator data in our clinical trials, na?
yeah, but in my company where I'm working, so there are clinical trials where my drug is not yet approved from other company also. So they are using as a comparator.
No, not used. So how can that be a comparator if it is not marketed? It is a
I don't know, but
compassionate use. It is a compassionate use or expanded access program. That is not a comparator, right?
so it might be like it is a supported trial or something like that, right?
See, wherever you are directly involved, means either you are supporting them or means you have the data for that, whatever drug is outgoing from your hand. So in that case, you can provide the sales data, you can give that data. But if you don't have control and somebody is doing on his own, then how can you provide that data? You can't chase that data, right? Because you don't have any records for that data. So in that case, we cannot provide them. Yeah. But in this case, if you are saying that my drug is not approved in certain countries and it is still being used, then that is a compassionate use program. So you as an MH are aware about that. You are providing them that medicine to be used for compassionate use or expanded access programs. Like suppose my drug is approved for NSCLC in US, but in Canada it is not yet approved for NSCLC. But I can provide the molecule or my drug in Canada also to be used as compassionate use or expanded access program. So that data can be provided, presented under the sales section in that you have other post authorization use, expanded access programs and compassionate use. In that you can provide that data. But if you don't have control, then why should you provide that data? You're not responsible for that.
But Amit, that drug is our company's drug, so it is our responsibility how it is circulating in the market and everything, right?
Suppose you are a MH, I am a distributor. Okay. You supplied me the drug because I am your distributor. Now your SAP sales, in your sales record, what you will give? You will say that I gave 100 packs to Fala distributors, means suppose Amit as a distributor. Okay. So your SAP sales say that 100 packets gone out of the company to the market. Right? Now, being a distributor, your sales data is showing 100 packs. Whatever is out of your company, it is there in your SAP sales. So when I will do post-marketing sales, I will consider that I will take it into account. Okay. Now, being a distributor, I gave 10 packs to or 90 packs to patients and 10 packs to some investigator who asked me. And now that investigator is doing some clinical trial, comparator trial, which you being a MH not aware about and you don't know, but your sales is 100, which is in their post marketing, not in the clinical trials. That sales is coming under post marketing section, right? But the use of that drug is not up to you. So that's why that post authorization use is there. Now, if that investigator is doing some trial and it is using as comparator or something your drug, and there is some adverse event reported, or it is reported in a database, some patient has something and it is reported in the database as off-label use. So that case will come to your line listing. So that case will be presented under post-authorization use. Your sales of 100 packs will be presented under sales section of post-marketing, but you are not aware about any comparator or any compassionate use program. So you can't provide the specific data of that compassionate use program. Being a MH, you are meeting your regulatory obligation. You are providing whatever sales is there. But since you are not aware what the investigator or what is being done in the market, you are not in the control of that, how the drug is being used in the market until and unless it is reported to you. In this case, the off-label use was being done, that was reported to you, then only you have been able to identify that the drug is being used as off-label and you are presenting that data in other post-authorization. That's why that post-authorization use section is there to present this information.
So if,
But
also
for
if other
sales, you cannot.
company is using it in a clinical trial and if they are not reporting you any adverse event or anything, that we can't help it, right? Like whatever.
No, that is their responsibility. That is their MH obligation to report to us. If they are not reporting, then what can we do? That's why these obligations are there for each MH. Na? If you might be getting cases of certain molecules which you are a manufacturer, but that case was received by another MH. They forwarded that to you, right? Then
Yes,
you
yes,
processed it
correct.
into your database. So that is a MH obligation. So they are reporting you, then only you come to know. In the same manner, for clinical trials, also it happens for the compassionate use or for this investigator initiatives sponsor or some other companies if they are initiating. And if some other MAH is initiating, then you should be knowing about it because they will report it to that CT guidelines is there, right? clinicaltrialsgov.in data website is there because they will provide their protocol to the regulatory authority or a health authority to get approval. In that, they will present the comparator information and everything. So that is how it comes. If you are aware, then you can present that data. If you are not, then you are not responsible if somebody is not telling you. Any other question, people? If no question, then probably we should close this call today. We are
So
to
Amit,
go quiet.
what is our plan from here regarding the training? So how are we?
Plan means
You are starting batch 2 as well, right?
not now. That's why I
So
told them that I'll announce the date on 1st January because I'm looking as
uh
to how I can complete this. And there is no overlap of batch 2 with you. Don't worry about that. There is certain different plan for them.
okay, that was my concern. Thank you.
Yeah, and I believe all of the safety sections are covered by now, right? Any section you feel that it is left for safety from the safety sections that are left?
So we are presenting clinical literature.
So
So
that
this
are clinical
section,
part. Safety sections, literature, section 11. I have explained certain things, but still I will take one more section on literature because that is also most important. So section 11 I'll take, don't worry. And then clinical trials. So section 11, I think whatever are left other than 17, 18 can be covered in one class. So probably the next class will be for those. And then one session on section 17 and one on section 18. I think 3 or 4 classes you people will be done with the PBRER.
Amit, there are 20 sections, right? Whatever we need to know about interview, we will cover it in next to four class. So all 20 section won't we cover, but whatever we need for
Everything
interview
is
will be
covered,
covered.
na? By that time, everything is covered. If we are covering 17, 18, then that is the last section that I was to cover. So all 20 sections will be covered by then.
So till section 4, we have like we were gone chronologically. 1, 2, 3, 4. Then we started on section 15, 16, right? Then section this medication
Five.
9.2, medication error. Then
9
section
itself, you
5,
can say 9.
9, yeah. Then 5.2, 5,
5.
section 5. Then next will section 11 and
Now
9.
next will be section 6, 7, 8, 10, 11, 12, 14 at once.
At
All will
once.
be covered in one
Okay.
section.
And then 17, 18.
And then 17, then 18. Probably
Okay,
17,
so
18 together.
and last executive summary will be in 17, 18 wala section only.
Executive summary is nothing. It is just a copy paste of the below section. So I'll tell you as to how that executive summary is written. You don't have to evaluate anything. Whatever data you have provided below, that is only copy pasted there. If you
Also,
see
Amit,
executive summary,
go ahead.
if you see executive summary of any PBRER, na, so you will see that they have the data from below sections only. It is only a copy paste. Nothing new is written there. Yeah, tell me.
So so this will all be covered. So PBRER will be completed. So can we have an overview of DSUR in any one of the session?
Overview of DSUR, PBRER, that will be in one session that I'll plan. Then there will be one session on signal evaluation report. 2 things more apart from this PBRER that will be covered. DSUR will be just be an overview. PBRER will be an overview in that. And then one session on signal evaluation report after these PBRER sessions.
Sir, means 1 more month we will take to complete.
4 classes, at least 1 month. But why you people are asking this question? Because till now, whatever is covered, I think that is sufficient to qualify the interview.
Yeah. Actually, I was asking because in one of the session you said that even though mainly the main questions are from the safety sections only, but we should know something about each and every section, like
Yes,
some
that will
overview should be there.
Yeah, so that's why I told you, it will be covered. Don't worry about it. But most of the things I think that you are asking, I thought that you are asking because you are not able to face the interview or you are not applying. So now you can apply because you can see your colleagues are now getting placed also. So most of the things, whatever is asked in the interview, 95% you have covered everything. Now 5% for suppose if they ask for 17, 18. Okay. So you can say that that is a part of medical affairs. From 16.5 on, like for 16.5, I told you effectiveness of, there is a medical affairs section. We don't write. Nowhere safety people write that. So it's fine. And 17, 18 also, you can say that the medical affairs team write. Apart from that, everything you have covered, you can face the interviews now.
Also, Amit, can we have a sample or any molecules paper report from where we can like have a look at it and understand everything?
Okay, so whatever is there in open source that you also can access, right, in Google if you find. And now if you want me to make you available, so can you tell me one thing? Could you be able to provide any SOP for your client to me? Any SOP of your client which you are working right now?
No, no, no. I will
Can you
not be
provide
able to.
that SOP? Any chance that by any means you can provide me the SOP?
SOP of what? Like
Any
for
chance or any trick? SOP of your client.
no,
Suppose the ICSR case processing SOP of your client.
no.
So
It
how
is
can you
strictly
expect
confidential.
me to provide so how can you expect me to provide a PBRER of my client or any of the other clients that I have worked?
No. I
Is it
understand
possible?
that, but no. I'm asking
So
about
that's why I
if.
am writing all those in. So that's why I'm writing all those things in introduction template so that you can have a rough idea as to what is presented in those sections. Then one of your colleagues only provided me the Vaxzevria paper, so I have forwarded you to you people because that is publicly available. So in the same way, I told you to go to EPAR website to have the RMPs which are publicly available. So try to find the papers which are openly available or Google or somewhere that you can find. But nobody can give you their client's paper. That is the risk of their job, right?
Right.
Sir, also generally means which paper we write for
Now
the
this,
PhD?
just a minute, let me pause the training because it will become a